So what is it doing to the cell, the overall cell, whenever it shifts from blue to red? How could you explain the cell to me?
So the first kind of stepping backwards is first we kind of need to know where that light is coming from. And that's been the big question. There's a strong hypothesis that the internal light, the unstimulated light, which is what we call biophotons or UPEs, ultra weak photon emission, there's a strong suggestion and strong hypothesis that they come from mitochondrial activity. So we call them reactive oxygen species. So when cells flux the electrons, they pull off the electrons from the food that you eat into your mitochondria, and they eventually end up in oxygen. That's what helps create the ATP that fuels our body. In that process, we create these things called reactive oxygen species. It's that produces the light. So is when the formation of ROS, you get this change from high energy to low energy that produces these lights. So the couple things happen. ROS are not so good for our cells. They produce damage. They're highly reactive, is a reactive oxygen, which is going to want to pull electrons from all of your cells. So the that's what we know. In terms of what this means for our cells, that's something that we're trying to figure out. The big question, I think, what we need to do is kind of blend in the biology of molecules. How do they actually interact with the light? And how does that affect the whole cell as a whole? We see this in photobiomodulation. We see that, certain receptors are sensitive to light based on the intensities and energies, but we don't really understand the whole signaling cascade at all.
Well, that's fascinating too, right? Because melanin helps with the ROS situation, and melanin's obviously absorbing,
Mhm.
like, especially neuromelanin, with it being such a black substance, is absorbing all of the frequency of.
Yes, absolutely. There are other proteins within our body that absorb light, right? Like I said, like every protein can be excited, right? With enough energy that it can be excited and it can emit light. And that's kind of what we hypothesize is happening in our brain. The biophotons that are being generated from the mitochondria aren't just random. They're used in some form of stimulation or communication with other proteins, but we need to figure out how that happens.
Is the ROS and this light being created in the mitochondria, or suspected to be created? Is that the things like the FO head spinning? And is it like the TCA cycle spinning, catabolic, or maybe even anabolic? And well, that brings up another question. Is there different light emission from catabolic to anabolic, even?
That's clever. I don't know. I mean, we know that during TCA, the whole point of TCA is to pull the electrons from the carbon that's coming in, right? The carbon source, the glucose in some cases, the proteins in some cases. That's the goal of these steps. Eventually, they end up in the electron transport chain where that electron kind of moves through it to get to its eventual home of oxygen. Um, that oxygen is what's going to help create this reactive oxygen species. So I'm not sure if within the TCA there's going to be much light generation. It's most likely going to be later down the line in that process.